what mean by totipotency and morphogenesis. This is the third lecture you will find here. As I said, the certain terminology explants means any plant organ or exercise plant part to initiate the tissue cultures. Now what is growth? Do you know what is growth? We are increasing in the size. That is growth? No. Yes. And increasing in the size due to the cell division and subsequent enlargement, that is growth. Now differentiation. The development of cells, tissues with the specific functions or regeneration of organs or organ-like structures or pro-embryos, that is called differentiations. Where it occurs, so the phenomena of mature cells reverting to meristematic state and forming undifferentiated callous tissues is termed as de-differentiation. Now what is redifferentiation? The redifferentiation means the ability of component cell of callus to differentiate in the wall plant or plant organ is termed as redifferentiations. Now we will see what is callus and cellular totipotency. Now callus may be defined as unorganized mass of loosely arranged parenchymatous tissues which develop from the parent cells due to proliferation of the cells, that is callus, mass of loosely arranged or parenchymatous tissues, unorganized mass of. Okay, that is called callus. As I said previously, cellular totipotency means the capacity of cell to give rise to a whole plant is known as cellular totipotency. Generally, a callus phase is involved before the cell can undergo and redifferentiation, leading to a regeneration of whole plant. Now, as I said, redifferentiation and de-differentiation. Cells can rarely give rise to a whole plant directly without intermediate callus phase or direct regenerations. That growth and differentiation alone proceed together, they are independent. Now, differentiation may be categorized into two groups. One is structural, another is physiological. The structural differentiations, it is further distinguished to external and internal differentiations. Now, external means most common example is root and shoot differentiations. Another familiar example is vegetative and reproductive phase of life cycles. Further, you will find in the male and female organs, that is reproductive organs. Okay. Now internals, these include the differentiation of all different various types of cells and tissues. Differentiated cell mostly occur in the group forming different types of tissue, vascular tissue. Now physiological means as the variation in the structure between root and shoot are expressed of fundamental physiological differences. As I cytodifferentiations, de-differentiation, cytodifferentiation. Okay, in both plants and animals, specialized cells perform different functions. This specialized is known as cytodifferentiations. Cyto means cells, cell differentiations in the plants and animals you will find here. The cell in the callus always you will find parenchymatous tissues or mass of cell or parenchymatous in nature. The differentiation of these cells into a variety of cells is required during redifferentiations of the cell into all plants. This redifferentiation of cell is known as a cytodifferentiation. Okay. Vascular tissue differentiations, that is, there are two examples. One is xylem and another phloem, food channels and nerve channels. Okay. Then organogenic differentiations means the generation of the whole plant from cell, callus, tissue or cytodifferentiation is not enough. There should be a differentiation leading to a shoot, but an embryo formation. This may occur either through organogenesis or it is called as somatic embryogenesis. Now what is organogenesis? Organogenesis refers to the process by which the explant, tissue, cells can induce to form root and the shoot and even whole plants. In other words, you can say the formation of organs is called organogenesis, that may be categorized into two groups. One is rhizogenesis or coelogenesis or cheilogenesis. The cheilos, the process of shoot initiation means coelogenesis, and rhizogenesis means root formation, the process of root formation. Now, what is the somatic embryogenesis? Somatic embryogenesis means development of embryo from the somatic cells. Okay, in the culture, the structure is similar to the zygotic embryos found in the seeds, and which is analogous embryonic organs such as cotyledons or cotyledons leaves. What are the factors affecting cytodifferentiations? Phytohormones, sugars, then nitrogens, then physical factors, and these are the various things which includes the factor affecting cytodifferentiations. Phytohormones, hormones here O. Okay. Auxin at low concentration stimulate xylogenesis, cytokinin and gibberellins. Also stimulate a tertiary element differentiation. When auxin and cytokinins or kinetin are used together, they have a synergistic effect. Sugar, what is the role of sugar? Sucrose play an important role in the vascular tissues differentiation, and it directly affects the relative amount of the xylem phloem formed in the cheilos. In the presence of low concentration of auxins, 1% sucrose induces the xylem formation. Better xylem differentiations with little no phloem were observed was increased 2% of xylem and phloem differentiations. Nitrogen presence of ammonia and nitrate as a nutrient medium we are adding. Then physical factors, the light, temperature. Okay. So the effect of light on vascular tissue differentiation varies between the culture tissues, and then temperature also different effect on cytodifferentiations. So the temperature, it should be within the 17 to 31s, promote the vascular differentiation. Besides the light, temperature, other physical factors that may affect your growth of the media, that is the physical factors such as pH of the medium greatly affect the cytodifferentiation, that you have to remember.